All right, so this is for 9.2 for biology. Picking up on cell reproduction, talked about the cell cycle. Now we're going to talk about the details of mitosis, interphase, and cytokinesis. So this first picture, I threw this one in because it's just beautiful. This is a karyotype. A karyotype is a term that has to do with genetics. And what it is, it's a picture of the chromosomes within your cells. And so humans have 23 pairs of chromosomes, so 46 total. And a karyotype lines them up in size order with the sex chromosomes, or the chromosomes that determine the gametes, at the end. So pair 1, 2, etc. Now, these are referred to as homologous pairs also, which we'll get to. So mitosis, the replicated genetic material needs to divide, and the cell needs to split into two. And this DNA, these chromosomes, need to separate accurately for it to work. So why do cells divide their nuclear material? Why do cells divide? Well, one reason is when you were born, I'm guessing you weren't as tall as you are now. Okay, so to get bigger, to attain mass, we need to increase the number of cells in a multicellular organisms like ourselves. As we grow, we need to add cells to make our bodies bigger. So that's why, one reason why mitosis. Another reason is to replace damaged cells. Remember in the last video I said red blood cells have a lifespan of 120 days. You need red blood cells to live, so the damaged ones need to be replaced. Or if you break a bone, there's another example. Here is a cell. This cell is in interphase. Now on your paper, it might need to be darkened in. Okay, we're going to add some information here. Some things you can see. This is a cell. Yay. This is a cell that is in interphase. All right, so circle interphase or write it. If you can't see it clearly, we can clearly see the plasma membrane, okay, on the outside. We also can clearly see so this is our plasma membrane. We can clearly see the nuclear membrane. Within the nucleus, in here, we have DNA. And the form of DNA, or the genetic material that we have here, is chromatin. So the relaxed form of DNA. That's interphase. Remember, that takes the longest. The next picture here, this is a cell that's in prophase. It's the longest stage of mitosis itself, prophase, P-M-A-T, PMAT. The chromatin is no longer there, and we see chromosomes. Okay, underlying chromosomes. In prophase, we see chromosomes appear. And within these chromosomes, the middle portion is referred to, and I'm going to add it here. And the middle portion, right in the middle, is referred to, that wasn't a very good middle there, boom, boom, as a centromere. Add that word. Okay, the middle of the chromosomes. And that middle connects what are called the sister chromatids. These are the legs of the chromosomes. So if I were to draw a chromosome, here's one leg, and there's the other leg of the chromosome, okay? Each leg is referred to as a sister chromatid. And each leg contains identical copies of the DNA. And they're attached, these legs are attached to the centromere in the middle. Also in prophase, you'll notice something is gone. We still have a plasma membrane, but our nuclear membrane is gone. Okay, and we also see these spindle fibers show up. I'll just show you in this last picture. In interphase, we didn't have that. So we see spindle apparatus. What's called the spindle apparatus, which is the centrioles plus these spindle fibers, the two centrioles. Prophase, okay, we see, oh, that's on the next page, good. Spindle fibers, centrioles, okay, appear and form what's called a spindle apparatus. And this spindle apparatus will attach to the chromatids, sister chromatids. Metaphase is the second stage, and it's indicative, or, well, I should say, it's clear that it's in metaphase because the chromosomes line up at the middle. Okay, right at the middle, it's called the equatorial plate. Okay, so right down middle. The next phase, anaphase, look at that. The chromatids are being pulled apart, okay, by that spindle apparatus, okay, and the spindle fibers shorten. So the sister fibers or sister chromatids separate, and they're moved to the opposite poles of the cells. That's during anaphase. Lastly, it's telophase. We can see that the nuclear membrane reforms, so write down 2 new nuclear membranes begin to form. The spindle apparatus disappears. I don't see it anymore. And lastly, you could write down, look at this. What form of the DNA is that? It's not chromosomes. It's not sister chromatids. But yes, you're right. It is chromatin again. Then lastly, the cell itself has to divide, including the cytoplasm all around here. And this is called cytokinesis. Cytokinesis looks different in animal cells and plant cells. Okay, what I just showed you there was essentially an animal cell. Okay, so here is one animal cell and here is another one. Okay, we have our nuclear membrane, okay? So in animal cells, the microfilaments actually pinch off to form 2 cells. Like if I had a piece of taffy and I pulled it apart from each other, it's pinching it off, okay? And this little area is called a cleavage furrow, this little indent. In plant cells, it does it differently. The cells are not as flexible. Instead of pinching off, in plant cells are more boxy because of the cell wall, it forms just a wall called a cell plate between the 2 daughter cells, a cell plate. And that's the end of 9.2.